third step, cross hybridization of selected plants. Cross hybridization of the selected plants. Sir, what is the meaning of hybridization? What is the meaning of hybridization? In first year we read about this hybridization, crossing two genetically different parent plants to produce a new plant. Therefore, producing a new plant by crossing two genetically different parent plants is called as hybridization, right? Therefore, producing a new plant, how? By crossing two genetically different parent plants. Therefore, by crossing two genetically different parent plants, we are producing a new plant. What is this process called as hybridization? What is this process called as hybridization? This is called hybridization. For example, take a parent plant with genes capital A, capital A. Take another parent plant with gene capital B, capital B. Now you tell me, these are two alleles of one gene, right? It's homozygous. These are two alleles of one gene, right? Homozygous. Two alleles of one gene, homozygous. Two alleles of one gene, homozygous, right? Now these are two parent plants. Parent plant 1, parent plant 2. Now listen, sir, for example, capital A stands for bacterial resistant. Capital B stands for viral resistant. B stands for viral resistant. Sir, now it has produced a gamete, sir, by name capital A. It has produced a gamete, sir, by name capital B. If these two gametes are fused, what we are going to get? Capital A, capital B. Now you tell me, sir, capital A stands for bacterial resistant, capital B stands for viral resistant. It means now a single plant is made both bacterial resistant and viral resistant. Yes or no? This plant is made bacterial resistant and viral resistant. Sir, initially, it means before producing this plant, is there any plant having both bacterial resistant and viral resistant genes, sir? No. Sir, this plant is having bacterial resistant gene but not viral resistant gene. This plant is having viral resistant gene but not bacterial resistant gene. But this plant is having both bacterial resistant gene and viral resistant gene. It means a new plant is produced. How, sir? By crossing what? Two genetically different parent plants. Producing a new plant. How? By crossing two genetically different parent plants. This process is called as hybridization. What is this process called as? Hybridization. And this newly produced plant is called as what, sir? Hybrid. What is this newly produced plant called as? Hybrid. Is this statement clear? Right. Now you tell me, sir, why we have to do this hybridization process? Why we have to do this hybridization process? No plant is with all desirable characters in it. Is there any plant having all desirable characters in it? No. No plant is having all desirable characters in it. So by hybridization, what we are doing means we are combining desirable characters present in two plants into a single plant. It means desirable characters present in two plants are brought into a single plant. By which process? Hybridization process. Therefore, no plant is with all desirable characters. All desirable characters. No plant is with all desirable characters. Hybridization, what? hybridization. Hybridization brings desirable characters. Hybridization brings desirable characters of two plants into a single plant. Therefore, desirable characters present in two plants. What? Desirable characters which are present in two plants are brought into a single plant. By which process, sir? Hybridization process. By which process, sir? Hybridization process. Is this statement okay? In first year, we read right, hybridization in detail. I am not speaking about chemistry hybridization, sp, sp2, sp3. Right. Right, hybridization in first year we read right, hybridization in detail. Yes or no? What are the first steps, sir? Selection of parents. What is the second step? Emasculation. What is the third step, sir? Bagging. What is the fourth step, sir? Artificial cross pollination. After artificial cross pollination, we have to re-bag it. Yes or no? These are steps what we discussed in hybridization in first year. Therefore, once again you have to check that statements. Is it okay? Right. Therefore, hybridization, what it involves, sir? Number 1, selection of parents. Number 2, emasculation. Number 3, bagging. Number 4, artificial cross pollination. Number 5, rebagging. Is it okay? Right. Now listen here. Therefore, by which process, sir? Hybridization process. We brought desirable characters present in two plants into a single plant. Yes or no? This newly produced plant is called as what, sir? Hybrid. Now you tell me, sir, this hybrid is superior over both the parents. Yes or no? Hybrid is superior over both the parents. How you are saying, sir? Because it is having characters of both the parents. So hybrid is superior over both the parents. Superiority of hybrid over both the parents is called as heterosis. This also we read in first year. Superiority of hybrid over both the parents is called as what, sir? Hybrid vigor. What it is? Superiority of hybrid over both the parents. Superiority of hybrid over both the parents is called what, sir? Hybrid vigor. Hybrid vigor. Sir, what is another name for hybrid vigor, sir? This hybrid vigor is also called as heterosis, right? What is another name for hybrid vigor, sir? Heterosis. Sir, this term is coined by Shell. Scientist name. Who coined the term, right, hybrid vigor or heterosis, sir? Shell. Is these statements okay? Right. Therefore, sir, this produced hybrid having desirable characters of both the parents. So this is superior over both the parents. This superiority of hybrid over both the parents is called as a hybrid vigor or what, sir? Heterosis. Hybrid vigor or heterosis. Now listen this statement. Sir, if we make a cross, can we expect 100% a plant having desirable characters of both the parents, sir? No. If you make 100 to 1000 crosses, if you make 100 to 1000 crosses, you will get one plant which is having desirable characters of both the parents. Therefore, out of 100 to 1000 crosses, right, one plant is obtained. Very often more than one plant is obtained. Very often more than one plant is obtained having desirable characters of both the parents. Is this statement okay? Right? Now listen this statement. Out of how many 100 to 1000 crosses, out of 100 to 1000 crosses, write only one plant, only one plant, very often more than one plant. Out of 100 to 1000 crosses, right, how many plants? One plant. Very often more than one plant is obtained with what? With desirable characters. With what, sir? Desirable characters of both the parents. With what, sir? Desirable characters of both the parents. With desirable characters of both the parents. Is this statement clear? Okay, it means the obtained one is a recombinant. Therefore, out of 100 to 1000 crosses, how many recombinants we are getting? We are getting one recombinant. Very often more than one, having desirable characters of both the parents. Is this statement okay? Right? Now listen, sir, we are discussing cross hybridization, right? Sir, it is time consuming and a tedious process. What it is? Cross hybridization. Cross hybridization is what, sir? Time consuming. Time consuming and tedious process. Time consuming and what's our tedious process? What? Cross hybridization is a time consuming and tedious process. Sir, tedious means boring process. We feel bored. Sir, why we are saying that it's time consuming and tedious process, sir? Once you tell me. Sir, in hybridization, first we have to select the parent plants. Yes or no? We have to select the parent plants. For example, right, this is one plant, right, this is another plant. We selected these plants as parent plants. What are the second steps, sir? Emasculation. What are the second steps, sir? Emasculation. For example, this is male parent, this is female parent. Male parent, female parent. Sir, what is the second step? Emasculation. Sir, in emasculation, what we are doing? We are removing anthers. What are removed? Anthers are removed. From which flower? Bisexual flower. In which condition? Bud condition. From which parent, sir? From female parent. Therefore, from female parent, when flower is in bud condition, right, we are removing anthers. What is that process called as? Emasculation process. Is this statement okay? Right. Sir, what is the third step, sir? Bagging. Now, can we call flower of this female parent as a emasculated flower? Yes. That emasculated flower is covered with a polythene bag. All these statements we read in first year. We are covering it with a polythene bag. What is this process called as? Bagging process. What is this process called as? Bagging process. Now, what is the fourth step? Artificial cross-pollination. What it is? Artificial cross-pollination. Sir, this is which parent? Male parent. Yes or no? Sir, why we selected this as a right parent plant having desirable characters? Why we selected this as a parent plant having desirable characters? Now this is male, this is female. Yes or no? Now what we have to do? Artificial cross-pollination. No. We are collecting pollen grains from anther. From anther of which parent? Male parent with desirable characters. It means which plant is having desirable characters. That plant we selected as male plant, male parent. Yes or no? From it we are collecting what's our pollen grains. Therefore, we are collecting the pollen grains. What we are collecting? Pollen grains. We are collecting the pollen grains from anthers. from anthers of a male parent with desirable characters, and we are transferring that pollen grains where, sir, onto the stigma of female parent with desirable characters. Is this statement okay? Sir, for doing this, it takes a lot of time and we feel tedious. So we are calling cross hybridization as a time consuming and tedious process. Why we are calling cross hybridization as a time consuming and tedious process, sir? Because we have to collect the pollen grains. What? Collection of pollen grains. From where? Collection of pollen grains from anther of which parent? Male parent. With what? Desirable characters. With which characters, sir? Desirable characters. And we have to transfer these pollen grains where, sir? Onto the stigma of a female parent with desirable characters. That's why we are calling this process as a time consuming and what's a tedious process. Is this statement clear? Right. Therefore, this is concerned to what's a cross hybridization of selected plants. Now, go for number 4. Selection and evaluation of recombinants. Selection and evaluation of what, sir? Recombinants. Selection and evaluation of recombinants. Number 4, fourth step. Now you tell me, if you make 100 to 1000 crosses, how many recombinants you will get? Only one recombinant. Sir, how you are going to call this as a recombinant, sir? Recombinant means having desirable characters of both the parents. Therefore, first we have to check which plant is having desirable characters of both the parents. If a plant is having desirable characters of both the parents, then we can call it as a recombinant. Therefore, we are selecting the recombinant first. Is it okay? Clear? Right. For example, right, cross 1. I am taking what, sir? Cross 1. Sir, in this cross 1, right, in this cross 1, we got capital A, capital B, sir. Now, going for cross 2. Sir, in cross 2, in what, sir? Cross 2, right, I got capital A, sir. Next, cross 3. In cross 3, I got capital B, sir. Is it okay? Right. Now, see these 3 crosses. In these 3 crosses, right, in this cross, right, we got desirable characters of both the parents. Yes or no? We got desirable characters of both the parents. We got what? Desirable characters of both the parents. Therefore, it becomes what? Recombinant. Sir, is this a recombinant? No. Is this a recombinant? No. Therefore, when we make a cross, we are not getting all recombinants. Out of all these plants, first we have to select the recombinants. Sir, how you are saying that this is a recombinant? Recombinant means having desirable characters of both the parents. Is this statement okay? Right. Therefore, what is this heading? Selection of recombinants. Selection of recombinants. Right. Therefore, sir, recombinant. Recombinant having desirable characters. Having desirable characters of both the parents. having desirable characters of both the parents. Sir, this selection and evaluation of recombinants is a crucial step for success of plant breeding. This step is crucial for success of plant breeding. Which step is crucial for success of plant breeding? Selection and evaluation of recombinants is crucial for success of plant breeding. Now you tell me, sir, what is the meaning of recombinant? Having desirable characters of both the parents. Sir, on plant it is written whether it is recombinant or not recombinant. No, on the plant it is not written whether it is recombinant or not. Then how, sir? We have to check the characters that are present in a plant. It means we have to evaluate the plant. Yes or no? It means scientific evaluation is required. What is required here? Scientific evaluation is required. Scientific evaluation is required. What is required? Scientific evaluation is required. What is required? Scientific evaluation is required. Therefore, sir, selection and evaluation of recombinants requires what, sir? Scientific evaluation. Scientific evaluation. Therefore, scientific evaluation is required. What is required? Scientific evaluation is required. Sir, once a recombinant is identified, what we have to do, sir? Once a recombinant is identified, it is self-pollinated for many generations. Recombinant is self-pollinated for many generations until it reaches homozygosity. Until it reaches homozygosity, so that characters cannot separate in the progeny. Is this statement okay? Right. Therefore, recombinant is a self-pollinated. Sir, self-pollinated or cross-pollinated? Self-pollinated for many generations. How many generations? Many generations until what is reached? Until homozygosity. Until homozygosity or uniformity is reached, so that characters, which characters? Desirable characters, so that desirable characters does not separate in progeny. Does not separate in progeny. Separate, segregate. Is this statement okay? Therefore, listen. In progeny, the desirable characters should not separate. In progeny, the desirable characters should not separate. If desirable characters not to be separated, they should be in which conditions are? Homozygous condition. They should be in homozygous condition. Then how we will get this homozygous conditions are? By self-pollination for many generations. Therefore, recombinant self-pollinated for many generations until we get homozygous condition, until we get uniformity in the desirable characters. Once the desirable characters are made homozygous, the desirable characters won't segregate in the progeny. The desirable characters won't segregate in the progeny. Is this statement clear? Right. This is concerned to what, sir? Selection and evaluation of recombinants. Now go for last step. Test, release and commercialization of new cultivar. What is the fifth step? Selection, test, release, and commercialization of new cultivar. Right? Now listen here. Sir, here what we are doing, sir? We already got a recombinant, right? Now first what we have to do means recombinant is first grown in recombinant, right? First grown in, right, research field. Research, what's a research field? Followed by in farmer's field. Now listen this statement carefully. Sir, we got a recombinant, yes or no? Now we have to check whether this recombinant is having desirable characters or not when grown in the field. Is this statement okay? Sir, when we are growing this recombinant right in the field, whether the desirable characters expressed in that plant or not, we should get that. For knowing that what we are doing, we are growing this recombinant first in which field, sir? Research field, followed by farmer's field. Therefore, first grown in research field, then we are growing it in which field, sir? Farmer's field. Sir, grown in farmer's field for 3 seasons. 3 seasons in different locations, different places, different locations of a country. Different places, different locations of a country representing what's are all agroclimatic zones where usually that plant can be grown. Sir, what is the meaning for this statement, sir? For example, take rice plant. In India, right, in all states, we cannot cultivate rice, yes or no? In India, wherever we can cultivate rice, do in all that areas, right, climatic conditions are same? No. Having different, what's are agroclimatic conditions. Wherever rice can be grown in India, in all such places, right, now we are growing this recombinant rice variety. In which field, sir? Farmer's field. For how many seasons, sir? 3 successive seasons. 3 successive seasons. Is this statement okay? Sir, if the recombinant is a wheat plant, in India, wherever the wheat plant grows, okay, in all that areas, right, now we are growing this wheat recombinant. Is this statement okay? Right? Therefore, first recombinant grown in research field, then grown in farmer's field. When growing in farmer's field, the growth is for how many successive seasons? 3 successive seasons. Where we are growing it? We are growing it in different locations, right, representing all agroclimatic zones where usually that plant can be grown. Is this statement clear? Okay. After growing it in which field, sir? Farmer's field, right, we have to evaluate this recombinant in comparison with the best cultivar present in that area. Therefore, recombinant evaluated by comparing by comparing recombinant. By comparing what, sir? Recombinant. By comparing recombinant with best cultivar. Best cultivar in that area. With best cultivar in that area. Sir, this best cultivar available in that area is called as a check cultivar. Check cultivar. It is also called as reference cultivar. It is also called as what? Reference cultivar. Sir, we are evaluating the performance of this recombinant. Yes or no? To evaluate the performance of this recombinant, we are checking this recombinant, we are comparing this recombinant with the best available variety, with the best available cultivar in that right locality. That best available variety or cultivar available in that locality is called as a check cultivar or reference cultivar. Check cultivar or reference cultivar. Thank you.